So let's talk about cell wall inhibitors. Instead of memorizing antibiotics, let's think about them as iterations, as each new drug exists because the previous one had a weakness. So essentially, we're addressing a weakness that the last drug had. We can only be attacking a specific set of pathogens, and those are the ones that have cell walls. So you're either gram positive or gram negative. Gram negative have a thin cell wall, and gram positive have a thick cell wall. This cell wall is made out of peptidoglycan. So there's 3 steps to make a peptidoglycan wall. So first, we build the precursors inside the bacteria. Then a membrane transports these building materials outside to the cell wall, where the cell wall is going to be built. And then there's going to be extracellular cross-linking of the cell wall to connect them. The way that we connect these cell walls is with a transpeptidase named penicillin binding protein. It was named after the fact that penicillin binds to it to inhibit this transpeptidase. First, let's talk about the non-beta lactams. The non-beta lactams that we're going to be going over are vancomycin and bacitracin. Vancomycin acts by preventing the linking of the diala-diala. What exactly does this mean? Well, diala is short for alanine, and these amino acids are used to link peptidoglycan together to build a cell wall. So if it blocks one of these dialas, the next one can't grab onto it properly, and therefore the wall can never be created. So that's what vancomycin does. It binds to block it. But the problem with vancomycin is that it can't penetrate gram-negative bacteria. Remember, gram-negative bacteria have an outer membrane that is between the outside world and the cell wall. So vancomycin can't cross the outer membrane. Bacitracin, actually, the way that it works is that it blocks bactoferenol, which is a lipid carrier that transports peptidoglycan out to the membrane. So it literally blocks a transport mechanism that gets this peptidoglycan precursors out to the cell wall. So that's how bacitracin works. Now let's talk about the beta-lactams. All beta-lactams do the same thing at their core. They bind to the penicillin binding proteins, which is actually a transpeptide that links this peptidoglycan together. We have the penicillins. They bind to the penicillin binding protein. They're great for gram-positive, the strepto family, and treponema pallidum, which is syphilis. Their weaknesses, that they can't penetrate gram-negative very well. And you know what else is their weakness? Beta-lactamases break down beta-lactams, and penicillins just happen to be very sensitive to those guys. Essentially, penicillin works great until the bacteria learns how to break it. So now let's go on to our next category. We have the anti-staphylococcal penicillins. Here, we have the same intrinsic mechanism. It binds the penicillin binding protein, but now we're going to add a bulky side chain to protect the beta-lactam ring. What does this do? It helps add resistance to that beta-lactamase that was making the original penicillin sensitive. It adds some beta-lactamase resistance. It's great, and it's great for gram-positive. It still can't penetrate that gram-negative outer membrane, but at least we have some resistance to the beta-lactamases. I would still say that although it's stronger against beta-lactamases, I'd say it's relatively weak compared to the other antibiotics. We're going to get into some that are very strong, but nonetheless, they are more resistant to beta-lactamases than the original penicillins. The anti-staphylococcals have poor gram-negative penetration. The anti-staphylococcals, to know for now, this list is not exhaustive. There's many more, but for now, let's just use these examples: nafcillin, oxacillin, dicloxacillin. Okay, I apologize. I see that there's a typo right here. Oxacillin. Okay, so now let's move on to the next upgrade we can do to these penicillins, right? So now we're going to have the aminopenicillins. What are we changing about them? What's actually making these advantageous over the last two is that we've improved their hydrophilicity. Remember when I told you gram-negatives have an outer membrane? This outer membrane, funny enough, it actually prefers things that a normal phospholipid bilayer would not prefer. Instead of a normal phospholipid bilayer, it favors stuff that is nonpolar, hydrophobic to pass through it, but the outer membrane in gram-negatives is actually the opposite. It prefers polar, hydrophilic stuff. So what did they do to the aminopenicillin to increase its penetration in gram-negative? They improved hydrophilicity, so now we can cover gram-negative bacteria. We're still somewhat relatively sensitive to beta-lactamases, but at least now we can attack some gram-negative bacteria. And the ones to know are amoxicillin and ampicillin for now. Here's where we were able to expand the toolkit available to us when we're trying to fight these bacteria. The cool thing about aminopenicillins is that we can combine them with a beta-lactamase inhibitor. And the reason why is because now we have improved gram-negative penetration because of the hydrophilicity that's added. We recognize that it's still sensitive to beta-lactamases, but now we're going to address that sensitivity to beta-lactamases by adding a beta-lactamase inhibitor. So with amoxicillin, we can combine clavax, we pair it, and this is great. We're taking advantage of the fact that we can have deep gram-negative penetration, and now we have a beta-lactamase inhibitor to address the fact that this guy is very sensitive to beta-lactamases. And with ampicillin, we can combine it with sulbactam. Same principle that we do with amoxicillin. Ampicillin has great gram-negative penetration, but it's still very sensitive to beta-lactamases, so we're addressing that by combining it with a beta-lactamase inhibitor. So now we have the monobactams. Same principle. They bind to the penicillin binding protein. These guys are great for gram-negative, and they're safe as a substitution for a penicillin allergy. The bad thing about monobactams is that they're very specific to just gram-negative. They don't do very well with anything that's just not gram-negative. They only do gram-negatives, and they don't even do anaerobes. But they still have their value as we can use them for very specific instances. And the monobactam, as an example, is acitronam. Okay, so now we have the anthoceudomonals. Were actually created very specifically to address Pseudomonas aeruginosa, those side fact. They bind to the same penicillin binding protein. The only thing is that Pseudomonals, they have a special penicillin binding protein that has a different kind of affinity that the other antibiotics cannot tackle. And anti-Pseudomonals address that by being able to have affinity for that specific penicillin binding protein. Now, these anti-Pseudomonals still have the same affinity for all the other penicillin binding proteins that exist out there. These guys have great porin penetration. Porins are located on the outer membrane of gram-negative, so they're great to penetrate gram-negative. They have affinity for Pseudomonas-specific penicillin binding proteins, and they're very broad spectrum because of this, because they can handle all sorts of penicillin binding proteins. The bad thing about these guys is that they're very sensitive to beta-lactamases, but how can we address that? Let's combine them with a beta-lactamase inhibitor. So one example that I think is very important to know is piperacillin. So the beta-lactamase inhibitor that we're going to combine with piperacillin is tazobactam, okay? And I'm just going to use a brand name here for once. Piperacillin tazobactam is known as Sosyn. And the reason why I'm saying this is because it's a very classic example in hospitals. It has very good binding affinity to several penicillin binding proteins, including pseudomonal binding proteins. It has a very good spectrum because it penetrates both gram-negative and gram-positives. So combining this guy with a beta-lactamase inhibitor plus this insane affinity and penetration makes it very broad spectrum. So that's why Sosyn is used a lot in the hospital setting, especially when you're doing empiric treatments. All right. Now let's talk about these cephalosporins. So cephalosporins are divided into 5 generations because they themselves have undergone their own improvements to make them better. And a general rule is early generations are better for gram-positive and later generations are better for gram-negative. That's just a general rule. It has its caveats. So the first generation binds the same principle. It binds to penicillin binding proteins, right? It's good for gram-negative, and I would say it has good skin flora coverage. Typically the Staph family, so we have Epidermidis, Saprophyticus, and Staph aureus. So they're good for these kind of guys. They're very limited in the sense that they have very little gram-negative coverage. Only the PEC, which is what I call PEC, and it's the protease, E. coli, and Klebsiella. No anaerobic, and it's beta-lactamase sensitive. The ones that are important to know are cefazolin and cefalexin. Second generation still binds to the penicillin binding protein. All of these beta-lactams are going to bind to that penicillin binding protein. But it's good because it has expanded its coverage to a couple more gram-negative guys. So now we can add to that list the hens, right? So Haemophilus influenzae, Enterobacter, Neisseria species, and Serratia, right? So they have a better gram-negative coverage. And these guys have also the ability to tackle on a lot of those anaerobes. So why are they bad? It's because they have weaker gram-negative coverage than the first generation. But the ones that are important to know are cefoxitin, and we're going to move on to the third generation now. And we have the mechanism of action still binds to the penicillin binding protein. It has a major gram-negative power, so these guys can cover gram-negative very well. So it covers all of it. The hens PEC, that's a mnemonic I use to know a couple of the main characters of gram-negative species, hens PEC, Haemophilus influenza, Enterobacter species, Serratia, Proteus, E. coli, and Klebsiella. And these guys can cross a blood-brain barrier. It has a good volume of distribution. It's bad because they go back to not covering anaerobes. So far, only second generation covers anaerobes. They're bad for gram-positive, and the ones that are good examples are ceftriaxone and ceftazidime. I put this little P here because this third generation specifically is good for Pseudomonas. So it has affinity to that specific Pseudomonal penicillin binding protein. Now let's go on to the fourth generation. Still binds to the penicillin binding protein. Strong gram-negative coverage, strong gram-positive coverage, and crosses the blood-brain barrier. So this guy is pretty broad. This guy is pretty good at just hitting everything. Bad thing about it is the more broad spectrum you are, the more likely you are to disrupt the flora in the natural flora that the human body has. And there's going to be opportunistic pathogens that are going to take advantage of the flora being disrupted and use it to then take over. One of them being C. diff, for example. And a good example is cefepime. I put a P here because it also has the ability to cover that specific pseudomonal penicillin binding protein. And then last but not least, the fifth generation still binds the penicillin binding protein. It's good for MRSA. Fifth generation is really good for MRSA. It has good gram-positive coverage, but here you can see that the spectrum decreases a bit. So it's less broad spectrum. I would say it's more of a targeted therapy. So we have ceftaroline and ceftazidime can bind to that pseudomonal penicillin binding protein. So it's good for pseudomonal. Ceftazidime is also combined with a beta-lactamase inhibitor to help it out some more. So that tells you that it's somewhat sensitive to beta-lactamase. So we combine it with tazobactam. And then the big guys. These are the grenades. If you throw these guys into the mix, they're going to destroy everything. They're very strong. They bind to penicillin binding proteins. These are carbapenems. They bind to penicillin binding proteins. They have really strong intrinsic resistance to beta-lactamase. Alone by themselves, it takes a lot of beta-lactamases to destroy a carbapenem. So it takes a lot. Like the beta-lactamases have to team up on this guy just to destroy one of them, if you have to think about it that way. It's very good gram-negative penetration, covers Pseudomonas, anaerobes, but it's bad because it's like a nuclear bomb. The whole floor is going to be destroyed, and those opportunistic pathogens are going to take advantage of that. A little memory aid you can use to know the carbapenems is DIME, D-I-M-E, doripenem, imipenem, meropenem, and ertapenem. Very important. Ertapenem, specifically ertapenem, does not cover those pseudomonal penicillin binding proteins. Real quick, before I move on, an important thing about cephalosporin, and I'm pretty sure you started noticing as I was going through the generations, is generations 1 through 3, there's this cool distinction. 1 is really good at gram-positive, so you can see it's farther away. It starts decreasing its gram-positive coverage as you start getting closer to 3. And then, on the other end, 3 has really great gram-negative coverage, and the gram-negative coverage starts going down as you start getting closer to 1. An important thing to remember is that the second generation are really good for anaerobes, all right? So if I had to put it in a number line so you guys can, so it's easier to visualize, respectively, where do they stand in broad to narrow, or narrow to broad, this is how I would put it. Bactrotracin extremely narrow. Vancomycin and penicillins, antistaph, cephalosporin 1s, they're narrow, but not as narrow as bactrotracin. Aminopenicillins are moderate in relation to, this is relative. This is a list that's relative to one another, right? The aminopenicillins are moderate, monobactams, cephalosporin second generation, broad, anti-pseudomonals, cephalosporin third generations, a little bit more broad. Very broad, I would say cephalosporin fourth generation, and then the broadest are the carbapenems. No one's going to be broader than carbapenems when we're talking about cell wall inhibitors. Remember, these penicillins, they grew out of upgrades. Each step fixes a limitation that the step before had. So we're fixing problems that the original penicillin had. We're fixing gram-negative penetration, beta-lactamase resistance, and anaerobic coverage. An important thing is also that beta-lactamase isn't the only resistance mechanisms. There's all sorts of things, but we're only focusing mainly on the resistance mechanisms of cell wall inhibitors. There's also resistance mechanisms like for Vanco, where the bacteria will purposely switch one of the dialas for another amino acid so that Vanco can't bind to that and do its blocking. So now the bacteria has gotten ahead of Vanco to make sure that there is no inhibition.